Hello everybody, this is Dr. Christopher White, and in today's presentation we're going to be continuing with evolution. So this is Evolution, the Theory and its Supporting Evidence, Part 2. So in the previous lecture we looked at the origins of the theory of evolution, the idea of natural selection, the genetics behind it, and the effect of natural selection on populations. So in this presentation we're going to think about how we classify organisms, some of the predictions that we can make using evolution, some of the supporting evidence behind the theory of evolution, and the idea of extinctions. So we're going to begin by looking at cladistics and cladograms. So once we've gotten the handle on the structure of organisms and how they evolved, the information can be put into diagram form. So cladistics is a type of biological analysis that splits animals into groups using derived characteristics. So a derived characteristic is something that is distinct to a limited group of organisms. So it's some feature that an animal has which allows you to differentiate it from other groups. Now, cladistics will ignore what are referred to as primitive characteristics. So this is a characteristic that many organisms possess. So a good example of a primitive characteristics would be something like all the land-dwelling vertebrates have paired limbs. So we have a pair of arms and a pair of legs. Dogs have a pair of forelimbs and hind limbs. So this particular characteristic is actually pretty useless if you're trying to establish relationships, because all of the land-dwelling vertebrates, well, nearly all the land-dwelling vertebrates should I say, possess paired limbs. So features such as hair or a free-boned inner ear are derived characteristics, and these derived characteristics are also sometimes referred to as evolutionary novelties. So remember, this is a characteristic that's going to allow you to separate an organism from the rest of the group. So they are considered to be evolutionary novelties because only one subclade has them. That's the mammals. So all mammals will have a free-boned inner ear and hair. Compare that to a reptile, which doesn't have either. So obviously you can split the two into separate groups based on those characteristics. However, all mammals have hair and a free-boned inner ear. So if we're just trying to classify mammals, then using hair and a free-boned inner ear isn't going to work because all of them have that feature, and therefore it would be considered a primitive characteristic. So if we were actually trying to classify mammals, we would look at other characteristics such as the presence of shearing teeth, or whether the third toe bears most of the weight. So that would be something that hoofed animals like horses and rhinoceroses possess. So the types of primitive characteristics that you use are controlled by the size of the group you're studying. So by tracing the derived characteristics, the evolutionary novelties, we can separate organisms into nice, tidy groups. And that's the aim of cladistics. So the classic method of displaying this information is in a phylogenetic tree. So on a diagram, which normally looks like this, we have two axes. We have the horizontal axis and the vertical axis. In the horizontal axis, it represents anatomical differences. Okay, so as you move across, the organisms are split into different groups based on the variations in their anatomy, whilst the vertical axis is for time. So essentially, as we progress up the vertical axis, we are essentially moving forwards in time. So the branches of this diagram are based on ancestor-descendant relationships, which essentially are based on derived characteristics. But the thing you'll notice is they don't actually stipulate what the derived characteristic is. And so, for instance, if we look at a couple of clades here, so we have dinosaurs and birds. Now, when we look at dinosaurs and birds, they're obviously rather different looking. However, there are some similarities. They do have the same derived characteristics. So one of the things that a lot of dinosaurs possess, especially the raptors, which are a subgroup of theropods. So the raptors includes dinosaurs like Velociraptor and Utahraptor. So that's the group that birds are descended from. And one of the characteristics we see in these more evolved raptors and the birds is we see a fused clavicle, which is otherwise known as a wishbone. And so the fact that these raptors and the birds both have a fused clavicle, that is a essentially ancestor-descendant relationship based on a derived characteristic. So that allows us to realise that birds and dinosaurs are related to each other, so we know that at some point birds must have branched off the dinosaur lineage. So cladograms are similar to the phylogenetic diagram we just saw. However, it shows you the characteristics that we're looking at when it comes to splitting the organisms up into separate groups. So if we look at the diagram here, we'll notice that we have a range of organisms that have been split into distinct groups. These are referred to as clades. Now we can then split the clades even further into smaller groups, which we refer to as subclades. So let's begin by looking at the diagram, and the first feature we're going to look at is a vertebral column. So all of these organisms here, sharks, lungfish, placoderms, salamanders, dinosaurs, birds, opossums, horses and cats, they all have a vertebral column. They all have a spinal column. So we can put them all together on the diagram. So that is going to be the characteristic we're starting with. So as we move along the diagram, the next characteristic is a bony internal skeleton. Well, all of these organisms here all have a bony internal skeleton. Sharks, on the other hand, do not. Their skeleton's made of cartilage. And so because they don't possess a bony internal skeleton, they are taken off on their own to form their own clade. As we continue along the diagram, the next characteristic is 4 limbs with 5 fingers and toes. Well, salamanders, dinosaurs, birds, opossums, horses and cats, they possess that feature. The fish don't. And so we're going to have the fish, the fish clade coming up here, and then we're going to split it into 2 separate subclades: fish with lungs, which are going to be classified as lungfish, and fish with gills, which are going to be classified as the placoderms or placiderms, depending on how you want to pronounce it. As we continue along the diagram, the next characteristic is the amniote egg. So dinosaurs, birds, opossums, horses and cats all produce amniote eggs. Salamanders do not, so obviously they go off to form their own clade. The next characteristic is hair and mammary glands. Obviously we know that that's a characteristic for the mammals, so possums, horses and cats, they're going to continue along, whereas dinosaurs and birds, which are not mammals, are obviously going to have to split off. And as we come along this diagram here, we'll notice that dinosaurs and birds do not, birds have wings, dinosaurs do not, so that's going to be the characteristic we're going to use to separate the birds from the dinosaurs. As we continue along, the next characteristic is a placenta. Well, horses and cats essentially are placental mammals. Possums aren't, they're marsupials, and so they're going to split off into their own clade. And then we have 4 toes. Cats have them, horses don't, so that's going to allow us to separate those 2 species. And so, as you can see, the cladogram here has separated out these organisms into distinct clades, and we can see the reasons why they've been split into those separate clades. So it's a very informative diagram. Now the point at which lines intersect, which are marked out here by the red arrows, that's the position of the most recent common ancestor. So the more organisms you have on your cladogram, the more complex it's going to become. Not really a huge shock there. So let's think about interpretation. So here we have 3 different cladograms for the relationships between birds, dogs and bats. So these relationships will change depending on the derived characteristics we apply. So if we look at the top diagram here, A, we're going to start off with, well, all 3 of these animals, they're all warm-blooded, so that's going to be our first characteristic they all share. As we move along the line, the next characteristic is a winged forelimb. Well, clearly dogs do not have winged forelimbs, so they're going to come off and form their own clade here. Birds and bats do, so they're going to continue along. Then, of course, we have the 3-boned inner ear. Bats, as mammals, will have a 3-boned inner ear. Birds do not. So, what if dogs and birds appear more closely related? So once again, we start off with warm-blooded. Next is eye design. Bats have a very distinct eye design which separates them from birds and dogs. That's going to allow us to separate bats off along one lineage, whilst birds and dogs continue. And then of course we have canine teeth. Dogs possess canine teeth, birds do not, so the birds are going to go off and form their own clade there. The final option, C, is going to focus on evolutionary novelties like hair and the birth of live young, for instance. So what we're going to do is we're going to separate the mammals from the we're going to separate the mammals from the non-mammals. And so obviously once again, warm-blooded, but 5-nailed forelimb. Bats have a 5-nailed forelimb, so do dogs, so they're going to go off one way. Birds do not, because obviously the forelimb has evolved to produce wings. In terms of the next derived characteristic, we have carnassial teeth, if you're wondering what they are. If you've ever looked in a dog's mouth and you've looked at the molars towards the back of their mouth, you'll notice the molars are kind of very sharp and jagged. They're a very specialized type of tooth that's designed essentially to have a shearing motion, a bit like a pair of scissors, and they're called carnassials. And so dogs possess them, bats do not, and so that allows us to split those two into their own clades. And so, depending on which derived characteristics we're using, we will be shifting around the position of the clades. And so we always have to bear that in mind. Where an animal falls on the cladogram is at the discretion of the person producing that cladogram. So scientists use cladograms to solve a variety of problems. So for instance, we can look at cladograms and we can trace back derived characteristics, and if we do it, for instance, we can see that whales are related to even-toed hoofed mammals, so deers, hippos and antelope. They might not look it, but once you follow the derived characteristics back step by step, you eventually end up at a point where they have a common ancestor. So this means that if we do this kind of analysis, there's going to be some kind of similarity between these organisms. So one of the similarities we can look for is something called a double pulley ankle. So if we have a look at this picture here, what can we see? Well, here we have the ankle for a dog, and then we have the ankle for a range of hoofed mammals. So the thing you'll notice is you can see we've got the two lumps on the top and we have the two mounting points on the bottom. So these are mounting points for tendons. So in the case of the hoofed mammals, we clearly have 4 of them. In the case of a dog, you can quite clearly see we have the 2 on the top, but we only have the 1 on the bottom. So we have a pig here, a deer here, and then we have Pachycetus here. And Pachycetus, if you come over here, there it is. It's one of the very early whales. Well, should I say, one of the organisms that's eventually going to give rise to the whales. And so by looking at the ankle design for Pachycetus, we can see that it's closely related to the hoofed mammals, but it's distantly related to organisms like dogs. So if we look at modern whale fossils, do we see the double pulley ankle? Well, the answer is yes, we do. Okay, so here we have ourselves 2 extinct whales, and here we have a bone for a pronghorn. So we can see once again, here's the double pulley ankle. We've got the 2 mounting points at the top and the 2 mounting points at the bottom there. Same thing over here for the extinct whale, 1, 2 for the top, 1, 2 for the bottom, and over here, 1, 2 for the top, 1, 2 for the bottom. So we can see that they are definitely related to each other, and a cladogram would allow us to visually demonstrate this. So we can also see it if we were to look at this. This is Basilosaurus, so this is a type of extinct whale. And if we look carefully, and if we look at the hind limbs, well, if we look carefully enough, we will once again be able to find the double pulley ankle in these vestigial hind limbs, which are pretty small and nearly completely gone. So another good use for cladograms is we can use it to identify plants and animals that may produce helpful compounds found in less common organisms. So for instance, if you go into the rainforest and you find a plant and you suddenly find that plant has the capacity to cure some kind of disease, well then obviously that's something you want to try and get to the general public. However, let's say the plant in question is extremely rare. And so it's very, very difficult to be able to get enough of the plants to produce the medication in the kind of quantities you require. Well, what you would do then is you would use a cladogram and you would try and find the closest relative to that plant that you could. Then you could go to that closest relative, and if you're lucky, that closest relative will have the same compound. And so if you're lucky, the closest relative will be far more common, and as such you'll be able to use it to produce the medication in much larger quantities. So that would be another good use for cladograms. So what about cladistics for fossils? Well, cladistics is much easier for living organisms. So we have to take care with fossils. Excuse me. So number one, we have to discern what are primitive versus derived characteristics. And this is especially difficult when you have groups of fossils that have a pretty poor record. So, it's very easy to split up organisms if you have a really good collection of fossils of that particular organism. You can see all the characteristics it possesses. However, if you have a poor collection of fossils, well, then it's far more difficult. So cladistics obviously focuses solely on the inherited characteristics that's being passed on from the parent to the offspring, and as we've discussed, it mostly focuses on the derived characteristics. So paleontologists must be careful to avoid characteristics resulting from convergent evolution. So that's a similar body form reappearing throughout the fossil record because that particular body form is so efficient for a certain environment. And we discussed how ichthyosaurs, which were marine reptiles, sharks, which are fish, and dolphins, which are mammals, all have a very similar body design because that particular body design works really, really well in a marine environment. It does, however, mean that because evolution is going to keep bringing that body design back up because it's so efficient, it means you're going to have lots of animals that superficially appear very similar to each other. And so if you're not careful, you could accidentally end up grouping ichthyosaurs, sharks, and dolphins together as one group, just purely based on their general body design. So you always have to look very, very carefully at the organism you're dealing with. Nevertheless, cladistics is a powerful tool that has allowed us to elucidate the relationships between many fossil lineages, and it's extensively used by paleontologists. So, when we classify organisms, we tend to classify species using a nested pattern of characteristics. So, closely related species will share several characteristics, and distantly related species will share fewer characteristics. Common sense, really. So, the classification scheme is based on the work of Carolus Linnaeus, and the classification scheme that he came up with gave organisms a two-part name consisting of a genus and a species. So, for instance, coyotes are Canis latrans, and wolves are Canis lupus. So Canis is the genus name that shows they're related to each other, but the species name, latrans and lupus, shows they are separate organisms. So what we can do is we can work our way back through the name, and we can work out essentially where they fit within a family line. So the Linnaeus classification is a scheme which essentially is an ordered list of categories. So everything's put into a set order. So the categories that the present range from the most inclusive to the least inclusive. So the most inclusive group is going to be a kingdom, then phylum, subphylum, class, order, family, genus, species. And a species should typically only have one member in it. A kingdom could in theory have thousands or hundreds of thousands of members. So here's an example from the textbooks. This is for the coyote. So we've got kingdom here, species here. So remember, kingdom's going to be the most inclusive, species is going to be the least inclusive. So obviously kingdom is Mammalia, so that's multi-celled organisms, essentially that produce cells have cells of a nucleus, reproduce sexually, ingest preformed organic molecules for nutrients. So that covers pretty much all animals. So then we have phylum, Chordata, so that's animals that possess a notochord, so some kind of spinal column. Obviously, that's going to remove any animals that do not have spinal columns. Then we have subphylum, Vertebrata. So these are organisms that have a segmented spinal column. So obviously, that's going to get rid of a whole range of different fish. Then we have the class Mammalia, so that's warm-blooded vertebrates, so any kind of cold-blooded vertebrates. So think of crocodiles, for instance, would be removed at that point. Warm-blooded vertebrates with hair or fur and mammary glands. Then we have carnivora, mammals with specialized teeth for eating meat. So that obviously gets rid of the herbivores. Then we have the family, Canidae. So that's dog-like carnivores. So that means you can exclude animals like bears, for instance. Then we have the genus, Canis, and that's made up of closely related species. So things like coyotes and wolves are very closely related to each other. And then we have the species itself, which is latrans, and that consists of similar individuals that in nature can interbreed and produce fertile offspring. So we've gone from a very inclusive group up here to kingdom, and as we've gone from one stage to the next, we've been removing organisms until we end up with a group that just contains a single species. And obviously when we name our organism, we will be, in the case of coyote, it's Canis latrans, and that reflects the genus name, Canis, species name, latrans. So as well as coming up with a scheme for the classification of all animals, Linnaeus also recognised similar characteristics between the animals, so he was using these derived characteristics. Interestingly, his classification scheme was designed for species that he thought were specifically created and immutable, so he had a biblical view. However, once The Origin of Species was published, people quickly realised that these shared characteristics constituted a strong argument for evolution. So here's an example of shared characteristics. So here we have a system, once again, going from kingdom all the way down to species. So we've got kingdom, phylum, class, order, family, genus, and species. So we're missing subphylum on this particular diagram. But once again, we have the kingdom Mammalia, kingdom Animalia, phylum Chordata, class Mammalia, order Carnivora. So you'll see as we're going down, we're reducing the number of organisms. So in the class Animalia, we have grizzly bears, black bears, giant pandas, red foxes, squirrels, coral snakes, and sea stars. Then we have Chordata, organisms that have a spinal column. Sea stars don't, so they're removed. Then we have the class mammals, so warm-blooded animals with fur, mammary glands, etc. Obviously, that's going to get rid of the coral snake. Then we have the order Carnivora, so that's mammals which are designed for eating meat, so that's the squirrel gone. Then we have the family Ursidae, so that's the bears, so obviously that's going to get rid of the fox. Genus Ursus, that's going to get rid of the panda. And then finally the species is going to be Arctus, so that's going to be the grizzly bear. So as we've moved along, what we've done is we've taken organisms that have essentially, the derived characteristics are getting closer and closer and closer as we move down the diagram. And so this means using the derived characteristics, we can see that grizzly bears and black bears are going to be most closely related to each other. Then those two are going to be related to pandas, and then those three are going to be related to foxes, etc. It's not a particularly rigorous system, but it does give you a good general idea of the evolutionary relationships. So the classification is very widely used, but it is arbitrary. So when organisms are assigned to categories above the species level, they're put into groups that seem correct. And this means that it does not, in all cases, actually reflect evolutionary relationships. That's why I say it gives you an idea, but it doesn't give you, a definitive relationship. So scientists therefore frequently use evolutionary relationships or phylogeny to group organisms. So we need to remember that a clade is a group of organisms that include the most recent common ancestor. So the aim of phylogeny is to essentially split these organisms up into clades and those clades into subclades using the derived characteristics. So a well-constructed cladogram will show a similar nested pattern to the Linnaeus classification, which we were just discussing. However, it will be a little bit more robust, and it will show the relationships between the organisms more clearly. So what evidence do we have to support in support of the evolutionary theory? Well, Charles Darwin put forward the following evidence. The first was classification, then embryology, comparative anatomy, geographic distribution. Now, if you remember, Wallace, who was the other person that came up with the theory of evolution around the same time, he was working in Southeast Asia, and his theory of evolution had a very significant proportion of geographic analysis of the distribution of organisms. So he was looking at how the organisms were distributed from one river valley to the next. And so this particular point here would have been very heavily influenced by Wallace's work. And then, of course, the fossil record, although at the time of Darwin, the fossil record was still extremely limited. So Darwin had no knowledge of the mechanisms of inheritance, biochemistry, or molecular biology. Those are far more recent advances. So modern studies in these fields can be combined with a much improved fossil record, has convinced the majority of scientists that the available information supports the theory of evolution as the best model to date. But is the theory of evolution really scientific? Which is a very good question. So a theory can only be considered scientific if it produces testable predictive statements, so we can make hypotheses using it. So when we think about something like Einstein's theory of general relativity, so whenever NASA or the European Space Agency is building a telescope or a satellite and it's planning to observe some new feature in the universe, what it can do is it can use the theory of general relativity to predict what the telescope should see once it's up there and actually doing the analysis. And so what you can do is you can make the predictions using general relativity, you send the satellite up, you collect the data, and then you compare it to your predictions. And thus far, general relativity has proven to be pretty robust. And exactly the same thing can be done with evolution. So the nice thing about evolution is that it's a theory with a substantial body of evidence to support it. But obviously we also need to always bear in mind that nothing in science can ever be considered 100% proven. So theories must constantly be revised and occasionally replaced if needed. So if new evidence comes up that says that general relativity is wrong, well then unfortunately it's going to have to either be completely dumped or it's going to have to be adapted to take into account the new observations. So the predictive capacity of the theories is one of the most helpful things. So obviously, as we discussed, going back to Einstein and general relativity, it allows astronomical people like NASA to make predictions about what they're going to see when they do their analysis of things like distant galaxies, stars, etc. However, not all predictions need to be about future events. You can make predictions, then you can test those predictions using things like the fossil record. So this is especially true in the case of evolution because the timescales involved in evolution are often quite huge. So to make a prediction based on evolution and then wait to be able to collect the data may well take decades, hundreds, possibly even thousands of years. So when it comes to making predictions about evolution, what we tend to do is we tend to make predictions and then apply those to what we can see in the historic record. So an example of this would be if wolves and coyotes are closely related, they should be similar not only in anatomy, but also their genetics, their biochemistry, and their embryology, so essentially how the fetuses develop in the womb. Now, if they didn't have strong similarities, then we would need to adjust or scrap the theory of evolution based on this data. However, when we compare wolves and coyotes, we see that they do in fact have very similar anatomies, they are genetically extremely close to each other, they have very similar biochemistry, so the chemistry of how their bodies function, and they have very similar embryology, so how the young develop in the womb. So that would suggest to us that they are closely related to each other. Obviously, the more distantly related they are, the greater the differences will be. So what predictions does evolution make? Okay, so prediction number one. The oldest fossil-bearing rocks should have the remains of organisms that are very different to those existing today, and the most recent rocks should have fossils similar to modern organisms. Well, that kind of makes sense, doesn't it? You would assume that modern organisms will be most closely related to the most recent fossils. And do we see that? The answer is a resounding yes. 2. If modern organisms descended with modifications from past species, there should be fossils showing characteristics connecting orders and classes. So this is the common ancestor. So what we should be able to do is we should be able to say, right, these two organisms are very, very close to each other, and we should be able to work our way back through the fossil record, and eventually we should be able to find a species that links the two groups together. And so as long as we have a good enough fossil record, we can do that, and we've been able to do it with quite a few different species. So for instance, we can do things like we can link together fish and amphibians from the Paleozoic, and we can work our way back through the family lines until we eventually reach a point where they converge on one species, which is the common ancestor. But remember, it's completely dependent on you having a good enough fossil record. Number 3. Closely related species should not only be similar in anatomy, but also in their biochemistry, genetics, and embryonic development. Distant species should show fewer similarities. That's true. So in the case of human beings, in terms of our anatomy, our biochemistry, our genetics and our embryology, we should show very strong similarities to chimpanzees. We will obviously be, but if we were to compare ourselves to animals like bears, we would obviously have greater differences in our anatomy, biochemistry, genetics and embryonic development, which would suggest that we are more distantly related. Number 4. So organisms descended from a common ancestor should show a nested pattern of similarities. Yeah, if you're related to, another organism and you have a common ancestor, you should be able to work your way back and you'll see the similarities, the body design getting closer and closer and closer until you hit the common ancestor. Number 5. Flora and fauna within caves should be similar to the flora and fauna found directly outside the cave, rather than being similar to species from other caves. And so this simply says is that if you have an isolated environment like a cave, the plant species you should see and animal species should be most closely related to the environment directly outside that cave. It's not going to be closely related to an environment, in a cave that's like, 100 miles away to the east. So, do we see that? The answer is yes. Now, this point is correct as long as the outside environment has not undergone substantial climatic change and that the caves are not interconnected. So if there's connections between the caves, well, that would allow plants and animals to migrate from one cave to another without actually having to, ever exit the cave, if that makes sense. So, this is true, but, there are some caveats that need to be put into the statement. Number 6. Land-dwelling organisms on isolated islands should most closely resemble organisms on the nearby continents rather than those from more distant continents. Well, that works very, very well for the Galapagos Islands. So we know that the finches of the Galapagos Islands are most closely related to the grass-quipped finches, which are found along the west coast of South America. So that shows that they're close together. They're relatively closely related in terms of things like their anatomy, their biochemistry, their embryology, etc. However, they're going to be more distantly related to finches from Europe, for instance, which obviously greater distance, greater adaptation to the local environment, and therefore the differences will be more extreme. So once again, point 6 holds perfectly true. Number 7. We expect mammal fossils to appear in the record long after the first appearance of fish. We would also expect reptiles to appear in the record before the first mammals and birds. Do we see that? Yes, we do. Number 8. When the fossil record for related animals, so something like horses and rhinoceroses, are examined, we should find they are similar and linked by a common ancestor. After divergence from this ancestor, they should become increasingly different. Do we see that? Yes, we do. So, on the whole, the predictions made by evolution are, to date at least, they match the evidence that we have available. So, just a quick point though, if humans were to disappear tomorrow, and in 1 or 2 million years a new dominant species was to arise, let's say a race of super intelligent mice, their theory on evolution would be slightly different to ours because human beings have had such a strong effect on the environment. So what we've gone and done is we've gone and transplanted multiple species into distant new environments. So that would make point 5 and point 6 a little bit more difficult to fulfill because human beings may have affected the flora and fauna outside the cave. And obviously, we've transported organisms into isolated environments that weren't there before. So a classic example would be something like pigs being transported to Hawaii. So this is a good place to stop. So get up, have a walk around, go and get a glass of water, take 5 or 10 minutes to relax, and then please come back for part 2.